Hello, everyone. It is my great pleasure to have a chance to present my talk here. My topic is bioinspired chiral supermolecular hydrogels. We know for hydrogels, typically during the last 10 years, it has becoming very promising materials to be used as biomimetic materials to study the life sciences. So here, I want to, for my talk, I have several questions. So the first one is, so what is the ideal biomimetic materials? This is uh, actually this is why everyone is working, why so many scientists are working with biomaterials, because all of them, they want to get ideal biomimetic materials. So from my understanding is for biomaterials, their final target should be biomimetic natural scaffolds, including their structure, property, and function should be very similar to our natural one. This is also the final. This is also the very challenge, of course, for man-made biomaterials. Therefore, I think before reaching the final target for the pupil, we have so many bottlenecks to be solved. Only this bottleneck to be solved, then can really have a natural, man-made biomaterials, their properties are very similar to natural scaffolds. From my study is I found one of very important bottleneck problem is for man-made biomaterials don't have chiral properties. This is a this is a very a big challenge as well, because since we know for all in our life, for our cells, actually they are surrounded by collagen or fibronectin and so on. Actually, all these kind of environments, they have these kind of materials, they have the chiral properties. So this is a very necessary properties for all the natural materials, but this is really missing, very important missing, critical missing parts for mining the biomaterials. So let's have a look why chirality is so very important. Because chirality is one of basic character of the natural biomaterials. I mean, they are related with most of biologic properties. So whatever the hard or soft materials, soft but natural materials, all of them actually are surrounded by the chiral properties, by the chiral structures, for example, protein. So what is chirality? Chirality is just like this. I will have these two small movies. You can see they have the helical structures. Yeah, basically they are same, but they are merosymmetric structure. So they're turning in different directions. This is left, this is right. So far, scientists already found a lot of disease are related with these helical structures. For example, Parkinson's disease, Alzheimer's disease. So all these are more or less related with the helical structure. So from this, you can see chirality is really very important property and also very necessary for our life to have a normal running. So for chirality, actually scientists already studied it from 1994. They realized at that time the cell growth or cell proliferation, they are quite similar. They are quite related with they have very big influence from the chirality, chiral properties. For example, the first example, they made chiral crystals. For the crystals, they have different turning directions. And they just put cells on it, and they found for cell adhesion behavior and cell proliferation properties a big difference. Unfortunately, at that time, it's limited by our characterization methods. So scientists cannot really have very deep analysis for this. Until 2007, the first research was started by New University from Professor Fuchs, and they made the films, yeah, layer films, two-dimensional films. On each films, you have left or right hand molecules, and then they just culture the same cells on both different chiral films, and they found on left and right surfaces and the cells, the same cells, they will have huge different adhesion behaviors. So this is from the chiral, from the molecular scale to study. But even though, because for chiral molecules, they cannot really mimic our our body structures. It's just from molecular scale, and then until 2011, and scientists they make the chiral, they use the chiral collagen. Of course, for collagen, natural ones have chiral structures. At the same time, they make artificial collagens. Here, they have mimic the chiral property. And as a comparison, also they make the fibers. These kind of fibers don't have the chiral property, but the dimension is quite similar as the collagen. And then they culture cells on these kind of films. Also is dimensional. This is on the molecular scale, but this is from the nanometers, from the structure scale. And then result is they found for the cells adhered on chiral collagen and on the mimic chiral collagen, they have the same adhesion or spreading behaviors. But on the fibers without chirality, you see the cells spreading behavior is quite a huge difference as the chiral collagens. So this is a very big progress. But the thing is, for this study, they cannot really to study the cell's behavior in 3 dimension. It's only 2-dimensional. Until 2014, the first paper to publish to study cells in 3 dimension. So what will happen for cells in 3 dimension with chirality? And then from here, we found for the left-handed nanofibers compared with the right-handed nanofibers, for a lot of cells, you read this paper, you found the cells, their proliferation adhesion speed is quite different. So this is really the first example to study cells in 3 dimension. So from here, we can know the cells has a very related with the chirality. So therefore, from here, we can see if we make the biomaterials, the better to have the chiral properties. Otherwise, their biomimetic property will be a little bit far away to the natural one. This is also why we need material to have the chirality. If we don't have chiral structure, what will happen? Then they have a limited biomimetic ability and a confined biocompatibility, and the biological properties will be influenced definitely. So what should we do? Since a lot of biomaterials so far, they don't have, man-made biomaterials don't have chirality, so what should we do? So before to think about this question, let's see what happens for synthesized biomaterials, why they don't have chiral properties. Because I'm working with hydrogels, I just take hydrogels to talk about these questions, hydrogels-based biomaterials. For generation 1, I gave 3 types, 3 generations for the hydrogels with the development of science. First one is natural hydrogels. You see they have microstructures with a very good chirality, of course, and biocompatible, that's no problem. But the thing is, for the study of the life science, for these kind of materials, their degradable speed is so fast. So we cannot follow long-term study. So because of this reason, scientists developed the second generation hydrogels, synthetic polymer hydrogels. For this kind of polymer gel, the advantage is they have controlled mechanical property. Yeah, you can to ensure you can have the long-term study of the cells. But the thing is, for this kind of hydrogels, and although they solve the mechanical properties, but they also lose their chiral properties. At the same time, for the polymer hydrogels, because they are very long covalent bonds connect to each other, they cannot really mimic our body natural or biomolecule behaviors. Thus, during last 20 years, the scientists developed a new types of supramolecular hydrogels. They have the physical interactions. They are exactly similar as our body biomaterials, and they have very good controllable self-assembly. This behavior is quite similar with biomaterials. The thing is, the matter is, for this kind of materials, the carrier property also cannot keep. This is also the big problem. Therefore, as I claimed from beginning, if we want to have gels, they can really mimic the biomaterials, natural biomaterials. We need to have hydrogels. They have controlled carrier property. So this is what should we do? So what can we do for this? Actually, this after long-term study from my points, I get inspiration from pyromagnetics. Everyone knows this phenomenon. For the first one, they have for these materials, every small units they have magnetic property. And the thing is, for all these units, if they pack very loosely, very disorderly from bulk, actually they cannot show any magnetic property because they are disordered. But if we align these magnetic units in one direction, yeah, with order become order, and then they will have much more stronger magnetic property. And also small magnetic units will have the big contribution on these behaviors. Yeah, from this I got the inspiration. So how can we solve the problem about the biomaterials that don't have carrier property? The thing is, what should we do is to let the molecules they pack very regularly, and then the carrier center we can transfer their carrier property to the whole bulk, and the final result is 1+1 will above larger than 2 definitely. Okay, so this is the inspiration. How can we get them very orderly packing? It will be the key. It will be the big challenge. How can we do them? Actually, the way to control them theoretically is very simple. To align the generators very regularly, yeah. Or you can use other external fields to induce clarity. So that's basically it's quite simple. But practically will be difficult. So what did we do? That will be our work. What did we do? Actually, I did it also from the supermolecular. Here, before do this, I must analysis for supermolecular hydrogels. Why they don't have the carrier property, even they have carrier center? And then analyze the hydrogelators for the traditional one. Actually, this is a traditional one. Normally, they have the structure. One part is hydrophobic, another part is hydrophilic. You can think about this kind of structure in solution, in aqueous solution, typically. Before self-assembly, they will form disordered clusters because the energy requires this. Otherwise, they cannot stay stably. So from here, maybe you got some you recall some of what I'm talking or what I talked from the previous slides. If these disordered clusters will be very bad for this assembly, because for this procedure will let all the carrier centers pack very disorderly. And then afterwards, even one small unit they have carrier center, and then afterwards for the whole structures, their carrier properties will be disappeared. So this is very important. How can we solve this? So from this, we need to have a new design for traditional generators. And then what we did is very simply, what we did, here is, it's hydrophobic, here is hydrophilic, and then I just add another hydrophilic part on this side. That's very simple change, but the influence is huge. This is whole structure, hydrophilic, and hydrophobic. You can think about these molecules. Before self-assembly, hydrophilic part can surround all the hydrophobic part. For these molecules, they have no chance to interact with other molecules. Then we successfully avoid this procedure. And the result is for these generators, they can directly self-assembly from single molecules to the nanostructures. If you recall the previous slides again, then these properties can ensure all the chiral centers, their property can transfer from single molecules to microstructures. Then you can get the whole structures with the chiral properties. So let's have a look at the results which we did. This is examples with molecules we did. Yeah, we have a binding center, we have chiral center. Here it is by complete pedal part. By changing the chiral center, then principle we can get left or right hand nanostructures. So this is experiments which we achieved with success. So we are very happy about these molecules. We got it. Yeah. Of course, for these supramolecular hydrogels, it's very sensitive to environments. So to be sure, we can have very highly sensitive assembly to get chiral centers actually for hydrophobic core center and hydrophobic arm. All of them you need to have a very careful design. So in these molecules, in these papers, we have very regularly talking about this. So for this slide, we want to tell you for our molecules, further proof why they can transfer the molecular chirality from molecule scale to nanostructure scale. Because in the middle we have the benzene. Yeah, the benzene actually they are hydrophilic. For all the water molecules, they cannot stay here. For chiral center, they are very close to this area. Therefore, the chiral center, they prefer to form hydrogen bonds between the chiral center. Water will have less influence on their self-assembly. This is also the one reason why our molecules can have very densely packing through the pyrostacking and hydrogen bonds. And as a result, they can transfer the property, chiral property from a molecule scale to the final nanostructures. This actually was recently published in last year. Another thing is for these molecules, the original molecules is this guy, left and right one. Okay, for these molecules, we have carboxyl acid group. Carboxyl acid group, this is a very useful group. So for this group, you can either covalently couple other functional group, also you can through physical interaction to get a self-assembly for the molecules and then to control their structures, chiral structures. The first example we did is by using the molecule to self-assembly with two unchiral molecules. And the result is the same chiral center, chiral molecules, and we use two unchiral molecules, we can control their chiral structures, left and right one. Okay, this is very interesting. Reason is for these molecules actually they can form extra hydrogen bonds. These extra hydrogen bonds have great influence on their chiral properties. Okay. Typically for this study also we found by changing the ratio of the chiral molecules, also you can control their self-assembly. You can get very good self-assembly molecules, chiral structures. And also we did another much simple one by using ions to co-assembly with our chiral center. And we found for the ions, with different ions, we can control their chirality very well by using this one. So all of these actually proof or precursor of these molecules, they have a very strong self-assembly property. Okay, you can control their structure very well. To further prove this system, we have our precursor have very strong self-assembly, and we co-assemble it with a coumarin. These coumarin molecules, they have fluorescence. Okay, so after co-assembly for these molecules, after co-assembly with all these with the precursors, you can get nanofibers with chiral properties, and also they called circular CPR, circular polarized light for these molecules. We can get without CPR, but after co-assembly, you can get CPR properties. Okay, so this is nanofibers co-assembly with coumarin, and this is CT signals. You see, we can get the symmetrical curves for this metal regulated CPL. And here I typically have very good strong G value. G value is to evaluate they have CPL light. Yeah, CPL light is very strong, very strong signals. Okay, so let's answer the final question. From above, as I talked about, the chiral structures is very important for the life. Okay, so what is the real difference for biomaterials with the chirality? Okay, so what we did is just incorporate the cells in different chiral hydrogels and study their chiral chiral structure influence the cell behaviors. And here we did actually in 2014, actually this already showed to you from very beginning, and we tested for more than tens of cells, and we found for all the cells almost they stay in left-hand nanofibers very well proliferated, very well compared with the right-handed one. It's 2 times higher even. So what will happen is, we did further study because we know the four cells actually they're surrounded by by the proteins. And we did mimic, we did the study of proteins interact with our chiral center. Actually, this is a movie. Yeah, you can see actually the proteins interact with our fibers. How can they interact it? Actually, during this procedure, they have some long-term selectivity to see which position can interact with nanofibers. This is the left-handed nanofibers, which not show right-handed one. Here, result is for proteins practically interact with the left-handed nanofibers, but not the right-handed. This is further proof for cell this phenomenon is quite similar with cell behaviors, cell interactive behaviors with nanofibers. The first proof our cell behaviors are regulated by the protein interaction with the nanofibers. Okay. Yeah, this is our first example to prove for the chiral center they really have influence on it. But the thing is that during the talk also we found the problem is for the above cell interactions we need to change the chiral center than to study the cell behaviors with chiral center. And the audience always ask us, so what influence from chiral center? Because you use left or right one to study the chiral structure influence on cells. And then my students have a smart design by using odd-even method. So what is odd-even method? It means the chiral center, the distance chiral center and the benzene in the middle, the distance. This is the methyl, the numbers of methyls. If they are 0, then you get right one. If they are 1, you get the left one. And again, they will get right one. It will be like this. This is the odd even. So here, this design, we will get rid of the influence with chiral center because here, left, all of them is left center. Just change this, just change the distance between the chiral center and the benzene, then you can easily control their structures. This is proven from SEM and from CT spectrum. Clearly see we can get that. And then finally, we started the cell behavior. What influence? Here from left-handed chiral center, we can get left-handed nanofiber, right-handed nanofiber. And the same by using right-handed chiral center, by using odd-even method, also we can get the left-handed, right-handed nanofibers, and then catch cells on it. Now we can compare these two, A and C. All of them actually they are left-handed nanofibers. Only thing is they have different chiral center. You have left-handed, right-handed, but we found the chiral center do have influence on cell behavior. For example, here we use left-handed chiral centers get left-handed nanofibers. The cells obvious it is much more compared with this guy. So from here we know for the cell behavior in 3 dimensions, not structures, but also their chiral center have great influence on cell behaviors. And finally, we did some stem cells to give a try. So what will happen for stem cells in chiral centers? And then later we found for left-handed and right-handed, they do have great influence on cell proliferation. Yeah. So for example, for left-handed hydrogels can enhance cell proliferation and differentiation into osteoblasts, but not for right-handed nanofibers. The further reason is the nanofibers, current nanofibers interact with the surface protein, and then they give a signal order to the nucleus and finally to influence their proliferation behaviors. So based on this, also we did some neutral differentiation of retinal progenitor, more or less stem cells. We did this work, and also actually the phenomena is quite similar as above, but the difference is for these kind of cells, red hand nanofibers will have more influence on it. So this is a very interesting observation. We are still working in this direction so far. So basically, this is the work we did actually from our group. Yeah, we did the very simply just design of keratinocytes and control their gelatinous structures, and then study the behaviors for cells with the keratinocytes. And then last year we have we were invited to write a review paper in Cons of Chemical Research, and this paper have very good summary for all the work we did during last 10 years. Finally, I would like to acknowledge my group members that all of this work actually did by my former students and also students now, the PhD students, and also our collaborators, yeah, in the world. That will be the talk which I gave today. Thank all of you. Looking forward to hear from you about good suggestions. Thanks a lot.